All right, welcome to The Explainer. Today we are diving deep into a science that pretty much makes all of modern biology tick. It's a field that had to be invented, literally, just so we could read the book of life.
So picture this. It's 2001. Scientists around the globe just dropped a bombshell. They finished the first draft of the human genome. And bam! Suddenly, we are hit with this absolute tidal wave of biological data. 3 billion letters of genetic code. Biology wasn't just biology anymore. Overnight, it became a big data science.
And that, right there, created a huge problem. Think about it. It's like being handed the biggest library in the universe, but with no card catalog, no index, nothing. How on earth were we supposed to read the book of life when we are literally drowning in its pages?
Well, the answer, as it turns out, didn't come from a biology lab. Nope. It came from something totally new, a field that was born out of pure necessity, right at the crossroads of two completely different worlds.
And that field is called bioinformatics. So, what is it? At its heart, you're taking the clean, logical power of computer science and you're applying it to the beautiful, chaotic, messy world of biology. It basically became the indispensable toolkit for wrangling all that new data.
And this quote just nails its origin story. People often call it a shotgun marriage between molecular biology and computer science. And it's perfect, right? It wasn't some planned, romantic union. It was two very different scientific cultures kind of forced together because the data was just too big for either one to handle alone.
Okay, so we know what it is, but what does a bioinformatician actually do all day? What's inside that digital toolbox? Let's take a look at one of the most fundamental jobs they have.
So a massive part of their work is something called sequence alignment. And honestly, you can think of it like a biological spot the difference game, but way more complex. By lining up these long strings of DNA or protein, scientists can spot regions that are similar. And those similarities, they can be massive clues about a shared ancestor, or even more importantly, a shared function. Now, there are a couple of main ways you can do this. You've got global alignment, where you try to match up two entire sequences from start to finish. But what if you're comparing, say, a human and a fish? Most of the code is going to be different. That's where local alignment comes in, and it's brilliant. It doesn't care about the stuff that's different. It just hones in on finding the most similar little pockets, these conserved regions that have stuck around through evolution.
The go-to tool for this, the classic algorithm, is called Smith-Waterman. It's a really smart process for sniffing out that best possible match. It basically works in 4 steps. First, you set up a scoring system. You get points for a match, lose points for a mismatch. Then you build this big grid to map the two sequences against each other. You fill in the grid cell by cell, calculating scores. And finally, you find the highest score in the whole grid and trace your path backward. That path, that's your best alignment.
So here's a super simple look at what that scoring grid, or matrix, might look like. The algorithm is building this kind of map of every possible alignment. It adds points when things match, takes them away when they don't. But here is the really clever trick, the secret sauce of Smith-Waterman. If the score in any cell drops below 0, it just gets reset to 0. And what does that do? It lets the algorithm completely ignore long stretches of code that don't match up well, so it can zoom right in on those little segments that are really similar. That's what makes it a local alignment.
But here's where it gets really interesting. This brand new field, remember, born from that shotgun marriage, almost immediately stumbled into this fascinating identity crisis. And it wasn't about the algorithms or the data, it was about the people.
Yeah, the whole debate pretty much boiled down to one single question. Is bioinformatics a real, distinct science in its own right? Discovering new things and pushing the boundaries of knowledge? Or is it just a technical service, a bunch of code monkeys providing tools for the real biologists in the lab?
And when someone actually did a survey and asked the community that very question, the results were just incredible. Look at this. The field was split perfectly right down the middle, almost 50-50. This wasn't some minor disagreement. This was a deep, fundamental rift at the very heart of the field's identity.
So to really get why there's such a split, it helps to think about the field in terms of 3 different generations of scientists, all defined by their relationship to that big Human Genome Project. You had the forerunners, who were around before it all kicked off. You had the founders, who were in the trenches building the field during the project. And now you have the followers, who came into a world where bioinformatics was already a thing.
And here's the kicker, your view on this whole discipline versus service thing probably depends a lot on which of those generations you belong to. The forerunners, who are trained as old-school biologists or computer scientists, they're way more likely to see it as just another tool in the toolbox. But the followers, the kids who maybe even got their undergrad degree in bioinformatics, for them, of course it's a science. It's what they've always known.
Okay, so identity crisis aside, where is all of this actually going? What's the endgame? Well, despite all the internal debates, the ultimate goal is crystal clear, and honestly, it's about to change medicine for every single one of us.
Remember that massive flood of data we talked about at the beginning? By creating the tools to not just survive that flood, but to actually navigate it, bioinformatics is the engine that's powering us toward a future of what we call personalized medicine. This is a future where treatment isn't one size fits all anymore. It's tailored specifically to your own unique genetic blueprint.
This incredible promise has a name: pharmacogenomics. And just think about the possibilities. We're talking about drugs that are designed to attack the very specific genetic weak spots of your cancer. Or how about a simple test that tells your doctor, before you even take a pill, whether you're going to have a bad reaction to it? Some people even imagine a future where we all carry around gene cards with our genetic profile, leading to an era of medicine that's truly, uniquely for you. And all of this brings us to one last really provocative thought. That shotgun marriage we talked about, it's not a temporary thing. It's a permanent partnership now. Biology is becoming completely inseparable from computer science. And in that process, the very definition of what it even means to be a biologist in the 21st century, well, that's being rewritten right before our very eyes.